Okay. Okay, so this is my screen, and I will start my presentation. So this is what I work on, sustainability of cycling in production with engineered microbes. My name is Effiong. My science group is group 47 in SynBio4ALL 2024 initiative. That's my interview. So the introduction for my project work, my project overview, the goal was to use genetically engineered bacteria to produce eco-friendly yeast. My objective was to reduce reliance on synthetic components and pollution from the production processes, because as we know, production process of ink involves and creates a lot of pollution, and that pollution creates a lot of health risks for humans that come from inhaling and digesting this and chemicals produced from the production process. Sorry, I didn't work on that part of my slide. The bacteria that produce the segment. So while reading up, I came across these bacteria that produce the segment. Then we have Chronobacterium violation, Serratia mixtesis, and Streptomyces collicolor, Pseudomonas aeruginosa, and cyanobacteria. Personally, I worked on cyanobacteria. The advantages and potential drawbacks, like most of the bacteria produce pigments that can potentially be used for ink, and some, like the first one, Chronobacterium violation, has and is said to have antimicrobial properties, but most of them are pathogenic, which creates unhealthy. And the aim of this whole project was to reduce health risks from pollution. We can't go about creating more in that process. So that was why the order in bacteria is here from number 1 to 8. From number 1, 2, and 4 were not included as bacteria that should be used for ink production process personally when this project pathogenic. Streptomyces collicolor and cyanobacteria, which is number 3 and 5, are the only ones that are not pathogenic. These two non-pathogenic bacteria produce variety of pigments compared to the rest that produce one pigment each. I think these produce about 3 pigments. The only challenge is their cultivation requirement can be very specific, which means you have to be very delicate when cultivating them. But however, then the bacteria has the advantage of being sustainable and eco-friendly because of their ability to utilize sunlight and CO2 using abundance. Types of pigment produced by cyanobacteria, as it was the bacteria that take for this purpose. We have cyclocyanine green, cycloerythrin red, chlorophyll green, carotenoid yellow to orange, and Idorenyin brown to black. Personally, I worked on cyclocyanine, which is blue. We used to into activity because my main focus for the e-production was for writing pens, because that is mostly used as a. And blue is the most popular color, so I focused on cyclocyanine. The disposal of all these pigments is generally environmentally friendly because of they have minimal environmental impact and they are very degradable and non-toxic. So traditional ink production and pollution impact. This is basically it. The production steps in traditional ink production, as they stated there. Then pollution sources, VOC, volatile organic compounds, can see examples of them. They are what we call air pollution, respiratory issues, and cancer that come from that accumulation, and heavy metals, which leads to water and soil contamination, which also further leads to health problems, and we have neurological damage. Yeah. Then we also have the waste that come from this production process that can also be quite damaging.
Yes. Then we also have the environmental challenges as a result of the whole health process. And this is why we need to tackle this problem with more sustainable methods. And what better method than synthetic biology, engineered microbes? So we have cyclocyanin production here. Yes, this was basically a picture to indicate cyclocyanin production. My proposed synthetic biology design was this. Yes. Sorry. My proposed synthetic biology design was this. First, the gene of interest, according to some research papers I looked through, was CPCA and CPCB, the alpha and beta subunits of cyclocyanin. Then the pathway overview, we have transcription from DNA to mRNA and translation into proteins. Then we have protein assembly, basically apocyclocyanin and the chromopore. Chromopore are basically what bring out the pigment in this they're basically what makes it gain color, basically. Then holoproteins, that is functional cyclocyanin with bright blue pigment. That's the final product. The associated protein in this pathway is cyclobilly which is protein that form these complexes. Basically, we can see then cyclocyanobili chromatopore attachment. So basically, the associated protein is cyclobilly protein. Then my proposed synthetic biology design was to engineer cyclocyanin production in E. coli instead of cyanobacteria. This would start by gene identification and cloning, and we already identified the genes as CPCA and CPCB genes from from the cyanobacteria. Then we optimize their codons. Then there's plasmid construction with suitable promoters. I suggested T7, which is an inducible promoter, and other regulatory elements. Then we ensure compactability and stability of our plasmid construct. Then there's transformation, which is the process by which the bacteria will take in plasmid, and expression. So the expression is prompted, like, how did I explain? It's increased using IPTG, which works well with T7, which was the promoter that I suggested earlier in the upper part of this slide, or any other inducer, depending on whatever promoter has been used to initiate cyclocyanin production. All this is just to upscale the expression and production of cyclocyanin. Then continuation of the engineering of cyclocyanin production in E. coli. We have optimization and production. Growth conditions also matter a lot, so they would have to be considered. And our inducer, we have to consider the concentration and the time to increase yield. Then that's basically it. We harvest and purify the cyclocyanin from, we harvest the E. coli cultures and extract cyclocyanin pigment from them. After the extraction is done, purification is done to get high yield of cyclocyanin. Then when we compare it with the traditional methods, we see that it has great potential to reduce pollution, which was the problem I was tackling through this particular intervention. We have the chemical use is reduced, all those petroleum-based pigments that are not really biodegradable, and all those harmful solvents used in the normal traditional methods are not used here because of it reduces chemical waste and pollution, as engineered E. coli approach mostly uses biological pathways. The environmental impact is reduced because of VOC. It minimizes VOC emissions and eliminates heavy metals. VOC emissions may still be involved due to final processing. I didn't really read up on that much because I can't really find much statistical data, so I couldn't include it here. But it eliminates the heavy metals involved in the production process. Then waste management is easier because E. coli products, like the ink being produced by E. coli, is biodegradable. And I would also suggest the use of glass pens for glass cases for the pens, because of glass is also biodegradable, seeing as it is made from sand compared to plastic, which would also align with the goal for ink production using engineered microbe E. coli in this case. The health impact, it reduces the health risks by avoiding harmful chemical exposures. As we see with the traditional methods, not only are workers exposed to VOC or heavy metals, but people living in the areas where such factories are situated, and the waste products that also get to the water can also circulate even farther than just the immediate area where these industries are located. And it promotes sustainable practices. E. coli production of ink is sustainable because of, first, it's biodegradable, and this bio-based methods basically simplifies the whole production process, making it a more healthier approach. Yes. And we still get our ink. So in the engineered microbe selection, I selected Escherichia coli because of its well-established genetic tool, rapid growth rate, and ability to express complex biosynthetic pathways efficiently. All those advantageous traits made me select Escherichia coli as the engineered microbe. And it is easy to cultivate. Its genetic makeup is well understood, allowing for easier manipulation, and a lot of tools have been developed for working with it, and it can express a lot of proteins and metabolites like covered earlier. All these traits support sustainable and scalable production of eco-friendly ink while aligning with our goal to reduce pollution and the health risks that come with the pollution. Pollution reduction calculation. This was just something I thought about, but seeing as I didn't carry out an actual experiment, it's just there as, let me say, some kind of guide for anybody who wants to do the experiment. There will have to be an optimization experiment and standard curve preparation and testing, and through that you can estimate the yield and you get the outcome, a quantitative outcome estimate for yield per liter of culture. So this is basically the goal. Using synthetic biology, we get bioremediation. Yes, basically, we can reduce the rate of pollution, which is a way of remedying the situation through biological means. And these were the references I worked with, apart from a few other sites I didn't reference here, but these were the two main ones that gave me insight while I was working on my project. Thank you. There's so much potential yet to be unlocked in the synthetic biology field, and we look forward to the growth of it all. Yes, so I'm basically done with my presentation, and I look forward to the meet-up question and answer session.